All right, maybe we'll get started. So it's a great pleasure to introduce our speaker today. First of all, it's good to see people. Luigi got his MD and PhD in Rome, Catholic University in Rome. He then did postdoctoral work in Chile as well as at Boston University, and then became an instructor at Harvard Medical School. He moved to the Department of Medicine in Madison in 2003 and has been affiliated with that department. He's now affiliated both with that and neuroscience. So Luigi has had a long-standing interest in Alzheimer's disease and has been working on that for many years. When his lab discovered the connection between the ER and acetylation and Alzheimer's disease, it led to their identification of acetyltransferase activity in the ER. And the rest of it is history. And I'll let him tell you that history. I just want to say one thing, which is that, you know, there are lots of little molecules, signaling molecules, metabolites, et cetera, floating around in the cell. And how the cell keeps track of this, how those molecules result in specific modifications only at certain places and at certain times in the cell is largely a mystery. Also, whether the cell, as it must, keeps track of sort of the global situation for this little molecule. And I think Luigi's work, the lab's work, is really, to me, one of the insights on where this is going, namely that the cell does obviously care, and these different little compartments and domains and organelles and stuff have to keep track of things, and that tracking has to be communicated cell-wide. And when it's not, you have disease. So, Luigi.
Thank you, Jerry, for a nice introduction. It's not easy to talk with a mask. I'll try to yell. I'm usually soft-spoken, but with the mask, I'm learning to yell. Although my kids know, they knew that I could yell on certain occasions. So it's nice to go back to in-person talks, presentations, to see faces on the other side. So there are 4 elements in this talk that I will discuss. First, proteostasis, which is a fancy word for protein homeostasis. Then metabolic crosstalk, and how the ER, the endoplasmic reticulum acetylation machine, actually connects this. So proteostasis, the cell makes proteins, and these proteins are made both on the endoplasmic reticulum as well in the cytosol. Some of these proteins will fall correctly, some will not. And so the cell needs quality control to select and sort them out. The correctly folded, they must be marked as good. And so they must be allowed to go where they're supposed to go and to do what they're supposed to do. The unfolded and misfolded polypeptides, they need to be degraded. First of all, they most likely are not going to be able to do what they were supposed to do. Second, they tend to aggregate, and therefore they can become very toxic. Metabolic crosstalk. I always say when I teach that my right hand knows what my left hand is doing, because the brain allows crosstalk between the 2 hands. So the same sort of crosstalk, homeostatic balance, must exist within the cell. So intracellular organelles and compartment must know what the other guys around are doing. So the ER needs to know what the mitochondria is doing, the mitochondria needs to know what the cytosol is doing, and so on. And so nature invented single metabolites, like Jerry already told you, that go around, okay, and transfer this signal and tell all the other organs what to do and what not to do. And so why the ER and why the acetylation machinery? So we stumbled on the ER. First of all, the ER is engineered to make proteins. About 75% of all mRNA and ribosomes are actually attached on the ER. And we know this since the times of George Palade. About half of those proteins will eventually be released in the lumen of the ER and they move along the secondary pathway. And the other half will be dumped outside on the cytosol. And this number obviously is different for cells and cells. Some cells depend heavily on the secondary pathway, and so they make more proteins within the lumen of the ER. Neurons, I had a neuron in the first cartoon. They need to ship molecules, vesicles, proteins on the other side. So polarized cells tend to depend on the secondary pathway more than fibroblasts, for example. So we stumbled many years ago, 2007 actually, we stumbled on ER acetylation in the lumen of the ER.
The sharing for team or for Zoom must have gotten turned off. If we just click on the Zoom and hit share screen.
Share screen, okay.
Oh, hold on.
No, what is, okay.
No, I think that's.
Post disable, but yeah. I need to become the host, so okay.
You are now co-host.
So share screen. Perfect. Okay. Let's get rid of this thing on the corner. Okay, we stumbled on the ER so many years ago. At that point, no one knew that there was actually ER acetylation, that there was acetylation in the lumen of the endoplasmic reticulum. And so we were the first lab in a paper in 2007 to publish that there was acetylation in the ER. Now, that gave us immediately several headaches. To acetylate the protein, you need three ingredients. First, you need the donor of the acetyl group, and that is acetyl-CoA. Then you need the acceptor of the acetyl group, that is a protein with the lysine residue. And then you need an acetyl-CoA lysine acetyltransferase, the enzyme that will transfer the acetyl group from the donor to the acceptor. And if you do have acetylation in the lumen of the endoplasmic reticulum, then all these three ingredients must exist in the lumen of the ER. Proteins is not a problem. The ER makes about 30 to 50% of all the proteins of the cell within the lumen. And so we have plenty of proteins. But first, the acetyl-CoA, the donor of the acetyl group, is generated, synthesized in the cytosol, highly charged, unable to cross a lipid bilayer. There had to be a transport able to shuttle the acetyl-CoA from the outside to the inside. And that led us to the characterization of AT1. Then there must be transferases that use the acetyl-CoA, and that led us to the identification and characterization of these two enzymes, ATS1 and ATS2. Type 2 membrane protein, you can see on this painting here in this cartoon, with a catalytic domain facing the lumen of the ER. And so we discovered the ER acetylation machine within 3 years, from 2007 to 2010. So AT1, the transporter, we did hardcore biochemistry and we discovered that it's a homodimer. And we discovered immediately that there were several mutations on this gene that were associated with severe disease. And I'll talk about that later on. They're all stop codon with the exception of two, these two, and they're all on the same loop. And so if you do the biochemistry, we discovered that that loop is important for the homodimerization of AT1. Two monomers, 60 plus 60, they join together to make the actual transporter 120 kD. And so that loop is essential. If you insert that mutation into a knock-in, for example, the mouse I'll show in a moment, or you knock in the cell, what you can see is that the prevents the formation of the homodimer and the transporter is inactive. Then we discovered that this transporter an antiporphyrin. That is quite interesting from the biochemical perspective, which means as acetyl-CoA goes inside from the cytosol into the lumen of DR, free CoA goes out through the same door. And that antiporphyrin mechanism provides the energy for this revolving door. And that is important for you see in a moment. Then we generated mice, and I'll show the mice in a moment. And what we discovered that if you have increased AT1 activity, there is increased acetylation. If there is decreased AT1 activity, there is reduced acetylation. The first part was quite interesting. If you have more AT1, more acetyl-CoA going to lumen of DR, you have more acetylation. Why? I mean, the fact that I have more bread in the house does not mean that I'm going to eat all the bread. So there must be a way for the levels of acetyl-CoA to activate the enzyme. And so there must be probably an allostatic way to regulate the ATAses. Then we start working on the ATAses. There are also dimers. We know there are homodimers. We don't know whether we can generate heterodimers, but we don't know whether heterodimers actually exist in vivo without transfecting the cell. And so 25 plus 25 is about 50 kD. That's the active ATA system. And we identify the dimerization domain. If you delete that, you prevent the dimerization and the enzyme is not active anymore. Then we did kinetics, the old-fashioned biochemistry, and what we found is that they use a bisubstrate mechanism, which means they bind both donor and accept at the same time. They have two pockets that bind at the same time, acetyl-CoA and the protein. And they actually now know that there's a channel that allows the acetyl group to be transferred from the donor to the acceptor. And that is important. And I'll go back to this later on. Interesting enough, these two ATAs are about 86% identical at the protein level, at the amino acid level, which means they are 14% different. The acetyl, they share basically an identical acetyl-CoA pocket, and there are three side chain the interactions that stabilize acetyl-CoA. And if you mutate that, you can block the acetyl-CoA activity, acetyl-CoA transferase activity. The interesting part is on the other side of the protein where the protein needs to insert, okay, the peptide lysine group. They are slightly different. The important thing here is that there is this lysine residue in ATS1 that needs to be acetylated. When acetylated, the pocket, the entrance for the protein that needs to be acetylated opens up. So an allostatic form of regulation. This allostatic mechanism does not exist for ATS2 because the opening is always, the pocket is always open. Which means when you have more acetyl-CoA, you have out-acetylation of ATS1, ATS1 becomes more active. And now you go back to what we found in mice, there was an allostatic mechanism to regulation. What does this mean? It means that AT1 can regulate the ATAses, specifically ATS1 through the allostatic mechanism, and the ATAses, however, can regulate AT1 through an antiporphyrin mechanism because they generate free CoA. If you have less free CoA, you have less AT1 activity. If you have more free CoA, the revolving door is going to speed up. And so nature designed this machine to be self-independent. Obviously, there are transcriptional mechanisms. We identify some of these mechanisms that allow to have more product of AT1 of the ATAs. But this is a system that allows the cell rapidly to modulate the acetylation machinery depending on the flux of acetyl-CoA. And so we go back to this concept later on. So proteostasis. Why this machinery and proteostasis? So it turns out that this machinery was designed by nature to regulate two essential functions of DR. One, engagement of the secondary pathway, and second, reticulophagy-mediated disposal of toxic protein aggregates. So what we discovered is that these ATAs work in collaboration with the oligosaccharide transferase, which attach the nascent oligosaccharide to the glycoprotein that is being made in DR. It turns out that these ATAs can only recognize correctly folded polypeptides. And so you have here a glycoprotein that has been made. It's correctly folded. It's recognized by the ATAs. It becomes acetylated. And because acetylated, now can leave the ER and go to the Golgi. If you mutate those lysine residue, if you change the structure of the protein to force the protein to become deacetylated, the protein is retained in the ER. Conclusion, the acetyl-CoA is a marker, a positive marker for correctly folded polypeptides, and regulates engagement of the secondary pathway, the ability of the protein to leave the ER and go to the Golgi apparatus. Now, what happens if the protein is misfolded or cannot be acetylated? It is not allowed to go to the Golgi apparatus and is retained in the ER. Obviously, that means it's going to aggregate and it's going to accumulate. And so the acetylation also regulates reticulophage-mediated disposal of this protein. And this mechanism took us a little bit more effort to characterize, and you can see there are many players here. First of all, nature invented the sensor of acetylation, ATG9A, autophagy protein 9A. Now, sorry, just want to mention, reticulophagy is a fancy word that means ER-specific autophagy. And autophagy is Greek for self-digestion, a mechanism to digest polypeptides or sick organisms. So ATG9A is a sensor, can be acetylated or not. If acetylated, is recognized by reticulate and is pulled away from FAM134B, which is a protein that acts as a receptor for the core autophagy machinery. And you can see this LC3 beta here. So if it is not able to interact with FAM134B, then there is no activation of LC3 beta and there is no reticulophagy. If ATG9A becomes deacetylated, is not recognized by reticulate. And as a result, it will interact with FAM134B, forcing a change in the conformation of the carboxyl end of FAM134B. Okay, and there is a protein HSPB1 that is necessary to stabilize ATG9A when it's not binding to FAM134B. The interesting part is that this mechanism is necessary to allow opening of a domain on FAM134B that recognizes LC3 beta, the LC3 beta interacting domain. And so you can see that the membrane will be here of the ER, so transmembrane domain, and then a part of FAM134B that goes inside. This bridge connects with this global part of FAM134B that has three domains made the same way, two opposite alpha helical structure with a coiled region. So alpha helix, coiled region, alpha helix. You delete this blue domain, and there is more activity, more interaction with LC3 beta. You remove the green motif, and there is increased activation. When you remove this fuchsia or whatever color, a violet color that I gave to this domain, there is a block in the activation of LC3 beta. And the domain that binds to LC3 beta is here. So it seems to suggest there is an opening mechanism. You need changing the folding and the structural conformation of this domain to allow this fuchsia or violet domain to open up. When it opens, now the leer, this yellow part, becomes available. And it turns out that this interaction, this opening mechanism, is recognized, is regulated by two calcium binding EF motifs that oppose each other, that ensure the closing and the opening mechanism. And indeed, now we're studying how calcium regulates this opening mechanism. And it turns out that calcium regulates induction reticulophagy, which we didn't know before this. So what we don't know yet is where and how reticulum binds to ATG9A. And that's what we're trying to dissect now. So the ER acetylation machinery regulates protein homeostasis or proteostasis within the secondary pathway by regulating engagement of the secondary pathway, as well as reticulophagy-mediated disposal of toxic protein aggregate. In conclusion, regulates the efficiency of the secondary pathway, the ability of the secondary pathway to make proteins the right way, ship them where they're supposed to go, and get rid of those that are damaged. Now, in this process, 10 or 15 years, we identify and characterize many proteins, AT1, ATAs, all these proteins. So you look at human databases, and what you find is AT1, homozygous mutation, heterozygous mutation of AT1 are associated with a specific form of peripheral neuropathy. Homozygous mutations, loss of function mutation, obviously, are associated with developmental delay and premature death. All these children die between the age of 1 and 5. Duplication of AT1 are associated with autism spectrum disorder, intellectual disability, progeria-like dysmorphia. So less AT1 is bad, more AT1 is also bad. Now, mutations of AT1, as I said, are associated with the peripheral form of neuropathy. And since, we'd like to complicate things, physicians, we give them strange names, and you can see many names, but they're all in the same group. They're all peripheral neuropathies. Then, mutation or disturbances in activity of ATG9A associated with peripheral form of neuropathy. Mutation in HSPB1 associated with peripheral form of neuropathy. Mutations in FAM134B associated with peripheral form of neuropathy. So it turns out that it seems that we have identified a molecular pathway that connects all these signaling molecules, these molecules, in the same machinery that seem to be associated with a similar human disease. And that is quite interesting. And so to address all this, first to analyze the biological impact of our discoveries, second, to understand a little bit more what this machinery actually does, we made a lot of mice. In 8 years, we made 16 mouse models. And I'm really tired of mice. But anyway, they're wonderful. It's a lot of money, but they're wonderful. Okay, so these are the mice that are associated with the acetylation machinery. First of all, homozygous knock-in that blocks the activity of AT1. These mice died during embryogenesis, day 7 or 10. Remember that homozygous mutation in humans are associated with premature death. The children die between the age 1 and 5. In the mice, they're lethal during development. Heterozygous mutation, knock-in again, in a mouse is associated with peripheral central form of neuropathy. Remember that the same mutation in humans is associated with peripheral form of neuropathy. But in the mouse is also associated, when these mice are exposed to pathogens, they are also associated with infection, propensity to infections, inflammation, and cancer. And you can immediately imagine how the autophagy process is essential for the formation of immunity and how the immunity requires secretion of immunoglobulins, so how this fits to the secondary pattern. Then we generated mice overexpressing AT1. We made neuron-specific overexpressors. These mice develop autism. Systemic overexpression, the whole body, they develop progeria, which is a fancy word. It's Latin and Greek, means in favor of old age. It's not normal age, the age we are going through. It's sort of a pathogenic form of accelerated aging. It turns out that duplication of AT1, as I told you before, in humans, it is are associated with autism, intellectual disability, and progeria. So the mice that we generate seem to recapitulate the human diseases, with obvious differences because these are mice and we are not. And so I'll show some of the figures now of this progeria mouse model. This is the guy with overexpression of AT1 in the whole body. They're born with the Mendelian ratio, but they're smaller than the brothers or sisters. Okay, these are litter mates. And you can see dysmorphic features. And obviously I had to trust the veterinarian on this because I don't really know how the mouse is supposed to, what dysmorphic means in the mouse. But you can see it looks really old at 2 months of age, okay? And the lifespan is quite accelerated. In 4 months, they're all dead. It turns out that they have all the features of normal or segmental progeria, okay? Short lifespan, females are not fertile, cardiomegaly, rhodokyphosis, osteoporosis, a series of skin issues, modest sarcopenia, severe rectal prolapse, and dramatic inflammatory response, infiltration of every tissue and organ, plus they have accumulation of senescent cells. So it's truly a model of segmental progeria. They cannot dispose of these toxic protein aggregates in DR. And so what happens is that you can see the morphology of the endoplasmic reticulum is changing rapidly. You have a large cisterna here compared to the wild type. You can see how large this cisterna-looking structure within DR. Not only the size has changed, but the morphology. You see the size is twice what you see in wild type, but the morphology has changed. All these vesicles are full of protein aggregates that cannot be released because the reticulopathy has been blocked. Now, these mice have overexpression of AT1, more acetyl-CoA. This acetyl-CoA is acting, is being used by the ATAses. And so the ATAses are the last output of this dysfunctional pathway. So in theory, if I take these mice, the AT1 mice, okay, and I treat them with inhibitors of the ATAses, I should rescue the phenotype. And so I'll go back to this issue of translation. But we dived immediately into this and went screening for compounds, and we identified one compound, which we called compound 9. And so we gave this to the mouse, 90 days old, the brothers or sisters with the compound. You can see they look completely normal. This is another mouse, 120 days old. This guy died, I believe, a week later. The brothers or sisters, completely normal. Lifespan, this works with males and females. They die fast with the compound that is complete rescue the phenotype. And so these ATAses appear to originate as a consequence of a gene duplication event. Lower organisms have one ATAse. We have two. You take Toxoplasma gondii or Plasmodium falciparum, and you shut down the only ATAse they have, they die. And so now I have people that work in malaria trying to figure out how to implement our discovery in their own model. And I'm shipping the compound to Australia. And I can tell you how painful that is. So why do we have two of these ATAses? 86% identical, 14% different. So we generate ATAse1 knockout and ATAse2 knockout. It turns out that they have a partially overlapping functions, but they're also slightly different. When it comes to DR, ATAse1 seems to be more important for reticulophagy. ATAse2 seems to be more important for quality control and macroautophagy. And remember that ATAse1 is the one that is allostatically regulated by AT1, by the influx acetyl-CoA. This tells me that my primary target for translation or in diffusion is ATAse1. And I'll go back to that later on. Now, I told you that this machine regulates each other. Acetyl-CoA goes inside, the ATAse is used, acetyl-CoA generates free CoA goes out. Now we're shutting down one of these enzymes. So technically there is 50% less free CoA being generated, and that is indeed the case. So what that does is blocking the antiporter mechanism. And so less acetyl-CoA is entering. And so there is accumulation of acetyl-CoA in the cytosol in both models. However, in the ATAse1, that excess of acetyl-CoA is basically mostly used for acetylation. In ATAse2, that excess of acetyl-CoA is mostly used to accumulate lipids. So the same molecule, somehow the cell knows that that molecule is coming from shutting down ATAse1 or ATAse2 and does different things. And that's what Jerry was also referring to, okay? So the cell knows more than we do, and somehow is able to discriminate the different pools of acetyl-CoA. Obviously, these differences are not absolute. So they have slightly different roles when it comes to reticulophagy. But as I told you before, at the beginning, these ATAses acetylate correctly for the polypeptide and therefore regulate the ability of the nascent polypeptide to leave the ER and go to the Golgi. So do they have different substrates? Do they acetylate different lysine residues? Now, all glycoproteins and glycosylation are glycosylated in the ER. The initial oligosaccharide structure is transferred from the donor, the dolichol pyrophosphate, to the asparagine residue. And it's this one. Then this protein, if is allowed to leave the ER, moves away from the ER, and as it does that, the oligosaccharide is trimmed down. For some reason, the cell decides to trim it down when the protein leaves the ER. And so you get these lower structures. And then the cell starts building up the oligosaccharide again when the protein reaches the Golgi. You can see it building up. Now, this is the most common structure of the oligosaccharide that you see everywhere in the internet. But the cell is always obviously smarter than the internet, and there's more structure than the internet. The important thing is that I can recognize these sugars. So you have fructose is attached on the cis Golgi. The transporter that transfers GDP fructose from the cytosol into the Golgi is in the cis Golgi. So fucosylation of a glycoprotein occurs in the cis Golgi. Then you have sialic acid. You have galactose first that is attached in the medial Golgi, and then you have sialic acid that is attached in the trans Golgi. So if I do use mass spec, I can actually look at the structure of this oligosaccharide, and I know whether the protein left the ER or not. I cannot discriminate between galactose and glucose, but I can see whether the protein has sialic acid or not. And so if the protein has fructose, I know that it reached the cis Golgi. If the protein has sialic acid, it manages to reach the trans Golgi. So if you do complex oligosaccharide glycoproteomics and you look at the structure of the oligosaccharide, what we find is that there is a decrease in the glycosylation in both knockout models. And you can see there is less oligoforms. This is cortex. You can't see because this thing is blocking, but this is the cortex. Hippocampus and female cortex, you can see male. So this is male, this is a female. Now, what is important here is that you can see 619, 129. I believe this is hippocampus then, and this is cortex. These are mislabeled. Anyway, one is cortex, and I believe this is ATS knockout. This is the ATS2 knockout. This is cortex of the males and females. So this is all females. You can see there are 619 changes, 129. So there is less in ATS2, more in ATS1. If you look at the cortex here, the males, you have a 180 decrease, 180 glycoforms less in ATS1 knockout and 153. It turns out that about 25 to 30% overlap we can detect on these oligoforms, which means that the other 70% oligosaccharides are specific, which means that these enzymes have shared substrates, but also very different substrates. And so now we generated mice with overexpression of ATS1 and ATS2. Again, we are brain people, so we like neurons, so we do everything in neurons first, but we also do systemic overexpression. And we're waiting for the results. I can tell you that the systemic overexpression have a quite severe phenotype, and so we expect to have very interesting results. What I told you so far is the protein is acetylated in DR, it goes in the Golgi. Turns out that that acetyl group, 2 carbonyl, is removed in the cis Golgi. So you have a protein that is 40, 60, 80 kD, it's a long protein. You have just a 2 carbon unit. The cell seems determined to remove that acetyl group. So what is the purpose of that? We believe we have the answer to that. But anyway, to be sure, we generated mice with knockout of the acetylase. And there too, we seem to have a very interesting phenotype. So now move to metabolic crosstalk. I already introduced the concept that when the ATAs are not working, the antiporter doesn't work, the acetyl-CoA accumulates in the cytosol, and you have changes. So when we did the mice overexpressing AT1 and the mice with deletion of AT1, we discovered there were dramatic changes in the levels of acetyl-CoA. I've been asked the same question for 70 years. Is this just the ER or outside the ER? And I always say it's just the ER. Turns out for those 7 years I was wrong. If you have an excess of acetyl-CoA, that acetyl-CoA is being used for something else, lipid metabolism and acetylation of proteins. And the opposite when you overexpress AT1. When you look at the change in the proteome, in the whole cell, what we see is change in the proteome and acetylproteome, which means some proteins go up as a result of more or less acetyl-CoA available in the cytosol. Some proteins will go down. Some proteins, the level don't change, the acetylation change. So the protein, the cell seems to change either the proteome or the acetylproteome to adapt. And those proteins are found in the nucleus, in the mitochondria, in the Golgi, in the cytosol, on the cell surface. So basically, the whole cell is reacting to changing levels of acetyl-CoA. For example, when there is less acetyl-CoA here in the cytosol, because I overexpress AT1 in the mouse, mitochondria start pumping more citrate out. It makes sense. The citrate is used by artery to make acetyl-CoA. So the mitochondria knows somehow that there is less acetyl-CoA and is pumping out citrate. So metabolic crosstalk. And so that led us to the discovery of a new pathway that connects the ER acetylation machinery, again, ER acetylation machinery, a machine that is in the endoplasmic reticulum, to these two proteins that are far away. One is on the mitochondria membrane and one is on the cell surface. They're both driving citrate inside into the cytosol. But in one case, the citrate is coming from the mitochondria. In the other case, the citrate is coming from the outside. Here in the cytosol, that citrate is immediately used by artery to make acetyl-CoA, and the acetyl-CoA then goes inside the ER. So I take the cells and I ask for a graduate student to do a simple experiment. Take these cells, overexpress 25A1, what happens? When we did that, the levels of endogenous ACTI went up, the levels of AT1, endogenous AT1 went up. Then we did the other experiment. We overexpressed 13A5. So that's the transgene that we're inserting in the cell. Immediately, the cell was able to upregulate endogenous ACTI and endogenous AT1. What does that mean? That the cell knows that there is more citrate, it immediately adapts. So we made mice. We made these new 4 mouse models. The neuron specific, we just came out a week ago or 10 days ago. Overexpression of 25A1 in neurons leads to autism. Overexpression of 13A5 in neurons leads to autism. Just like the overexpression of AT1. Duplication of AT1 in humans are associated with autism. Duplication of 25A1 in humans are associated with autism. Duplication of 13A5 in humans are associated with autism. So again, consistency in the phenotype, and the mice are able to mimic the disease in humans. And so let's see if this works. Okay, so this is one of the features of these mice. On the left, 25A1, on the right, 13A5. So this is during the day. So the mice should be resting or sleeping, and most of them, the leader mates, are resting. This guy who inherited the transgene instead has this repetitive hyperactive jumping. During night, again, these guys are active eating. The other two instead, who inherit the transgene, spend most of the time jumping. They also eat because the weight is not changing. And obviously, there are other paradigms that we use that we published. When you do MRI in the brain, you see changes in the myelin formation and the connectivity within the brain. There are changes in the diameter of the axons and the formation of myelin. So there are changes in white matter, which we detected here. We're able to confirm with electron microscopy. When you take brain slices in vitro, you see there are changes in synaptic plasticity, specific in long-term depression. When you look at the morphology of the spines, there are changes in the size of the spines. So everything seems to fit with the phenotype. Then you take these neurons, you play them, and you see their firing potential. They fire more than the wild-type neurons. Okay, so they have sort of hyperactivity. Then we need proteomics, and there are many proteins that change. They go up and down, and they all have to do with the same things. The machinery that makes the synaptic vesicles on the arm, pushes them through the secondary pathway, makes the spines on the surface, and regulates the activity of these spines, just like AT1. And we see this in the hippocampus or in the cortex. Then we did the acidoproteome, looking at the acetylation of this protein, and again, we see similar results. So again, we have identified molecular pathway, metabolic pathway that seems to lead to the same disease phenotypes. I have to say that there are differences between these 3 mice. 25A1 and 13A5 are more similar between each other, slightly different from AT1. And obviously, there might be some biochemical explanation for these differences, which we haven't tackled yet. And so now we're looking at the systemic. So far, all good. AT1 neuron overexpressors, 25A1 overexpression neurons, 13A5 overexpression in neurons, slightly different, but overall the same. Then we did 13A5, and we got into trouble. The 13A5 behaves like the AT1, progenitor. The 25A1, not so much. So again, the cell seems to know systemically outside the neurons whether this acetyl-CoA is coming from the citrate that is coming in from 25A1 or 13A5. And that's something that really stuck with me and gives me headaches at night. So again, a progenitor-like phenotype. You can see this guy looks really old and sick, die fast, and have the usual things that you see in progenitor. It just, I have to say, it's a bit more severe than the AT1 overexpressors. So we have identified a molecular pathway that connects all these ingredients that seems to lead to the same phenotypes. Translation. So the model here is that 13A5 is pumping more citrate. This citrate is being used by actylic acetyl-CoA, and this acetyl-CoA is going the lumen of the arc through AT1, and as a result, the ATACs are working at higher speed. So technically, if you shut down ATS1, the last output of this pathway, you should be able to rescue the phenotype of 13A5 overexpressed, if everything is correct. So we did that. This is wild type leader mate. These are the two mice. You give the compound, you can see there is a rescue of the phenotype. Now, maybe Jerry already recognized this. This gene was initially characterized in the fly, and it was called Indy. This guy, Indy, which is not, doesn't stand for Indiana Jones, is I'm not dead yet. It was a fly phenotype that didn't want to die. It kept living, okay? And so in the fly, downregulation of 13A5 leads to the Indy phenotype. I'm not dead yet. In the mice, the opposite, overexpression, leads to a progeria phenotype. Interesting, in the mouse, if we shut down 13A5, there is not much phenotype. So these genes have evolved differently. In humans, duplication are associated with autism and progeria, but mutations that block 13A5 are associated with encephalopathies, epileptic form of encephalopathy. So there are differences across kingdoms against this evolution in these genes. So this one always gives me trouble. Let's see if I can shut it down. I don't know which one. Okay, another movie where you have the wild type, 110 days, they look completely normal, leader mates. The 213A5, 90 days old, you can see they look very bad, very sick. You can see the other guy now can barely walk. The leader mates with a compound, they look completely normal. There is a dramatic rescue of the phenotype. So now I'll move to the last chapter, translation. I already introduced some of this translational effort, but now I want to introduce the concept. There are many diseases that are characterized by the aberrant accumulation of toxic protein aggregates across lifespan. And obviously, you immediately talk of aging, where it's very prominent, Alzheimer's disease, Huntington disease, Parkinson's disease, etc., cancer, nephropathies, chronic nephropathies, and so on. But there are also diseases where you have a mutation in a specific protein that leads to misfolding and aggregation of these proteins. And these are obviously early in the lifespan. Again, lots of neuropathies, but also disease outside the brain. So, and I've been talking about ER. So technically, the question is, can I actually target this translation, this machine, the ATAses, and rescue, help the cell dispose of toxic protein aggregates that form in the ER, not in the cytosol, in the ER, and therefore rescue all these disease phenotypes? And so I introduced the concept of efficiency of the secondary pathway. And so this concept of proteostasis within the ER, within the secondary pathway, is so important that the nation invented multiple complex mechanisms to ensure proteostasis. And defects in proteostasis are the basis of many diseases across lifespan, concept that I just introduced. And as a result, proteostasis is an active target of biomedical research. Lots of startup companies, in case you want to make your own. So we went on, we were able to build an assay to measure acetyltransferase activity of ATS1 and ATS2. We gave that to the facility at the Wimmer, at the Cancer Center. They optimized that. They screened the library of 15,000 compounds. And when we ended up with one compound out of 15,000. That's the compound 9. So we gave that to a mouse model of Alzheimer's disease, and there was a complete rescue of the phenotype. And the same paper here, this brain paper, we used 3 mouse models. Alzheimer's disease, APP is translated in DR, inserts into DR as secondary path. Correction of the phenotype. Huntington disease, the Huntington protein is translated in the cytosol. It aggregates in the cytosol. This machine should not work. No rescue of the phenotype. ALS mouse model, SOD is translated in the cytosol. Aggregates in the cytosol. This machine should not rescue the phenotype. No rescue the mouse model. So there was a proof of concept built within that paper. And then obviously the progeria rescue that I showed you before. The problem with that. So we published this paper. I got an invitation from the NIH, come and look at the workshop. And so I'm very excited. I go there. First talk, first slide. 95 to 97% of all compounds that succeed in mouse failed in humans. I was depressed right away. So we went from a library of 15,000 compounds, we ended up with one. And 97% of that single compound is predicted to fail. So we need more compounds. And so first we work to define structural properties of these enzymes, doing structural biochemistry. And so these on the left is ATS1, on the right ATS2. We know now there is an acetyl-CoA pocket. We know where the protein inserts. We know that there is a channel that allows communication between these two. We know that there are slightly differences in ATS2. And so now we went back, we said, well, can we use this tool now to define our screening process? And that's what we just published here. I think it also came out last week. A pipeline that allows us to screen these compounds faster and test them faster. And so first thing is, I have my compounds, and I selected the 30 most active compounds that came out of the 15,000 library screen. And so can we dock these compounds in silico, sitting in front of the computer? We can do that. Can we measure energy of displacement of either acetyl-CoA or the peptide lysine structure? We can do that. And by doing that, for example, if you look at acetyl-CoA, you can select some top candidate for ATS1 and ATS2. We also know that these compounds seem to go in different places in ATS1 and ATS2. So they seem to have different affinity and therefore energy, presumably energy of binding, with these ingredients, with this enzyme. And so then we ask the question, well, you don't really know how this compound is going to react in water, in the organism, the structure of this compound. There are going to be many orientations, and depending on that orientation, it's going to sit in one pocket or in the other. So can we predict the top energy orientation? We can do that, and that's handled E and F. Now, based on that orientation, can we select the highest energy? Okay, in these two enzymes, and we selected for 3 new compounds, and compound 9 is just our poster child because it's the one that we knew worked. And so we can do that. And by doing that, what we're able from these 3 compounds, so we're now to 3, we know that compound 10 and 19 are potentially strong inhibitors of both ATS1 and ATS2. But we also know that the compound 11 is not going to inhibit very well ATS1, but is going to inhibit ATS2. And so we measure obviously the Lipinski rule to see where these compounds were predicted to be absorbed, survive liver, metabolism, go in blood, and able to cross the blood-brain barrier. And so we use this as obviously as a proof of concept. The idea is to go forward and to have a larger analysis of compounds. So we need the perfect mouse model, and we have AT1 STG. In 2 months, that's sick. In 4 months, that's dead. So in 4 months, I know the answer. I don't have to pick a mouse model that lives 2 years and then wait 2 years to have that results. And so this is the wild type, 6 months, looks normal. This is the AT1 STG, winning. Starts with the diet. No diet, this is how it gets after a few months, okay? Compound 10, completely rescue the phenotype. Compound 19, completely rescue the phenotype. Compound 11, sort of mixed results. And you can see the lifespan, compound 10 and 11, which inhibit ATS1 with high energy, high affinity, I should say, behave like compound 9. Compound 11 that preferentially inhibits ATS2 has sort of a mixed effect. And so then we went from this compound to a mouse model of Alzheimer's disease. And we picked this mouse model, which has a more severe phenotype, the original APP mouse that we published in 2016. But again, we see the same rescue. And again, compound 10 and 19 are better inhibitors. 19 is actually better than 10. So I'll end the talk here. So in 2007, we discovered there was acetylation in VR. And now we went on to discover VR acetylation machinery. And we know that this machinery regulates 2 essential features of ER proteostasis and dynamic of the secondary pathway. One, engagement of the secondary pathway itself, And second is reticulometry-mediated disposal of toxic protein glycogen. But because it was designed, it also regulates metabolic crosstalk between DR and the other organs within the cell. And so this is the pre-COVID times when you can actually have people at home and have a nice Christmas party. And so this was the group. Hopefully we can go back to these parties soon. And you can see some new postdocs that just started in the lab. And I think I'll stop here. Thank you. Yes.
Great talk.
Thank you.
So I think Ellen was talking about the three compounds and compound 11 wasn't as potent and inhibitor. Would it be worthwhile to like try to modify the compound so that it can become more potent?
Yes, you can do that. Technically, you can do that. So what I'm showing that is a pipeline to identify more compounds. So that now I have not just one, I got more than one in hopes to move forward. Then obviously you have to do the usual thing, the PK, PD, see whether this is going to be toxic or not in humans. We've done some of this work for compound 9, and the results came out negative, which means theoretically it shouldn't kill any human. But obviously, you know, this is just the in vitro assay. It's not toxic, so worth continuing. But then obviously you can modify the structure to move forward and have your best candidate. Yeah. And the companies want that. Want that because if you have a natural compound, you make a certain amount of money. If you have your own compound, you make much more money. I don't know, I'm not after that, but you have to deal with these things. Yes.
So you need a really fine tuning of the level of API, I think, because the mutants showing the phenotype of overexpression also shows.
Yeah.
So that compound actually you give to the wild type may affect something or?
It does. It does. Some more, some less. I don't remember which one, but one of these compounds, for example, can cause steatosis. And you can imagine that. Not every single tissue goes through that, just steatosis, so just liver, okay? It's inconsistent, not every mouse develops, and so not every compound. So there must be some sort of form of regulation. But that's easily solvable because, you know, steatosis is not a big deal. You run a little bit more, eat a little bit less, and you're fine. And if you have Alzheimer's, you want to do that, yeah. Yes.
A bit more about how if a specific transmembrane protein is misfolding and therefore fails to get assimilated, how that would lead to ATG9 changing acetylation levels, and then also does the reticulopathy occur at a specific domain of the ER where the misfolding is occurring, or is it kind of indiscriminately eating up chunks of the ER?
I don't have the answer to that question. First of all, what we know, okay, these are two questions. I'll try to tell you what we know. If you take the protein that is correctly folded, becomes acetylated, you change the structural region of that protein, so you force the protein to fold in a different way, it is not acetylated anymore. If you change the lysine, but you allow the protein to fold correctly, it's not acetylated anymore. In both cases, the protein is not able to march to the secondary pathway. The ATAs are coupled with the oligosaccharide transferase. 25 plus 25, 50 kD. We pulled down the ATAs1, it came down with a complex of 350. Came down together with the oligosaccharide transferase. So they work together. What is the trick that nature invented there? The oligosaccharide transferase, the process is co-translational. It recognizes a specific consensus motif within which there is an asparagine residue that when it's 14, 15 amino acids away from the surface of the R, 40 Armstrong, is recognized by the oligosaccharide transferase and becomes glycosylated. Deacetylation is post-translational. There is no consensus motif. It can only occur if the specific structure is recognized by the ATAs. That's pretty cool, the way nature invented this thing. The regulation of reticulopathy, obviously we are limited from the system. by the system, okay? So what we can do is push this on the limit to the extreme, more acetylation or less acetylation, okay? So that's how we manage to build the reticulophagy pathway and system. Obviously, the question is how does that work in the normal situation, in me speaking here and you standing there where we don't have overexpression of AT1 or shutting down of AT1. That's difficult to dissect. That goes to the question how the non-acetylated goes to one pathway to the other. One thing that I failed to mention, or forgot to mention, is that when you have a misfolded polypeptide in the ER, the single misfolded polypeptide can be shipped through the ER membrane back to the cytosol, ubiquitination, proteasome. The protein aggregates cannot. Those are disposed of through reticulophagy. So what I mean by regulation of these two events is that you need the ER acylation machinery to work at a certain speed and efficiency, like Aki just said, so that these two processes are preserved. The individual proteins, how they work, when you can go one direction or the other, we don't know. The question of ATG9A, is this happening on the same in one specific place of the ER or not? I don't know. People pull down ATG9A from the autophagosomes. What did they get? A bunch of protein associated with autophagy. What we did is overexpress AT1 in this system, ATG9A, sorry, and then pull down ATG9A just from the ER, not of the autophagosome. Remember, ATG9A is in the ER. When activated, it goes to autophagy. The autophagy machine is already assembled. Okay. If we pull down ATG9A from the ER membrane, what do I get? I was expecting autophagy protein, 0. I got all the proteins involved in secondary pathway. All the proteins at the COP2 system, the SEC enzymes, proteins. So all those proteins that are essential to the formation of this exit site, move this exit site out of the ER and go to the Golgi. So this autophagy protein 9A seems to work with the two systems. Depending on the post-translation modification, can go toward autophagy or can just shuttle along to the secondary pathway. And there is a question that some students ask me, what happens with ATG9A supply of membrane? That ATG9A can go to the Golgi and then serves as supply of membrane for the autophagosome. So there must be, like you asked, going back to your question, there must be somehow a way for the ER to know whether ATG9A should go here and there, the specific location. There is crosstalk between the machinery that goes to autophagy and the machinery that goes to the secondary pathway. What determines whether ATG9A goes to one side or the other, we know it's a post-translation modification. But then is this happening where? I don't know that. Good question. Yes. Sometimes I can't give an easy question, I know, an easy answer. The things are always more complex than us.
I have a question.
Go ahead.
So in the situation where this trying to determine the source of the CO-CoA, the cell, some part of the cell, is there any chance that these different events occur at different times of day? And so the detection for one is at one time and the detection for another is at a distinctly different time?
Yeah, I would predict that's the case. We don't have any evidence to support that. We haven't studied that, but I would predict it's the case for certain cells. I mean, we already know that the ATAs and AT1 are regulated in a different way during memory formation, during response, certain response. And so makes sense. You're shipping more proteins on the cell surface, you're making memory, you need spines. And so there are differences. So one enzyme works different than the other one. You can imagine the same situation changing in pancreas, where it seems to be very important. When you have exocrine function needed, you are eating and need to digest, or when you're resting and sleeping at night and you're building this thing. So there must be a way to regulate the activity of this depending on the time of the during of the day, depending also the physiology of what you're doing. Like in the case of activity dependent, the activation of the ATAs is simply because we are talking and trying to memorize what I'm saying. Yeah. But that's tricky to dissect that.
Yeah. You have the biochemical.
Yeah, and we have the mice. Yeah, but still.
A highly motivated person.
Yeah. Postdoc or graduate student is willing to give me 10
Postdoc.
years of his or her life. 10 years, no less than that.
Any other questions? Okay, let's thank Luigi for a great talk.
Thank you.